So first of all, I want to thank the organizers for inviting me. It's a really lovely meeting and getting this opportunity to talk to you guys about this. As the chairman said, today I'm going to talk about a Luminex-based assay for diagnosing rheumatoid arthritis. And rheumatoid arthritis is a chronic inflammatory joint disease, and it affects the peripheral joints, and the symptoms are pain and swelling, and eventually this leads to a joint destruction and loss of function if it's not treated correctly. And it's roughly up to 1% of the population, predominantly women. And one of the probable autoantigens is type 2 collagen. And type 2 collagen is the main protein in the cartilage. And it's been shown that it's quite frequent in RA patients that they have both B cell and T cell involvement against type 2 collagen. And if you immunize mice or rats or humans, or also most likely humans, with type 2 collagen with an adjuvant, you will get a rheumatoid arthritis-like disease called collagen-induced arthritis. And this is the gold standard for rheumatological research in animals. And what my lab has been doing for many years is to characterize the specific epitopes on a type 2 collagen. And type 2 collagen is actually a Homotrimer, so it's a triple helical molecules. So it's a bit challenging to investigate this because you can't simply call up your local peptide chemist and say, hey, can you make this peptide for me? So you actually have to make a triple helical structure, otherwise you can't detect these antibodies. And there are only a handful of labs in the world that are able to synthesize these. And the region is very conserved. So we find essentially identical epitopes between both mice, rats, humans, and monkeys. So we can in great detail investigate these different epitopes and their different functions. And some of them are shown to be pathogenic, but also some of the antibodies towards some of these peptides are actually protective. And this is kind of the basis for our kit. And here's just some data showing when we used these peptides in RA patients, just an example of some major epitopes. And interestingly, if you add a citrullination, and this is a post-translational modification on these peptides, you get a much higher response in RA patients. So what is citrullination, you might ask? So this is a post-translational modification where the arginine, the amino acid arginine, is converted by the enzyme PAD in the presence of calcium to an amino acid called citrulline. And the important difference here is that the arginine residue is charged and the citrulline is neutral. And what happens is we have a different folding and it's more sensitive to degradation. So this is, one can call it kind of biomarker for inflammation in one sense. And it occurs naturally in the body, for example, MVP and filaggrin and histone proteins. And at sites of inflammation, also some other proteins can get citrullinated. So a major discovery made around 20 years ago was that antibodies towards citrullinated protein peptides are really specific for rheumatoid arthritis. So it's around 70% of all RA patients have these antibodies. And the specificity is somewhere between 95 to 99%, depending on the assay. And currently in the clinics, they use something called the CISP2 kit. And this is a Dutch invention, actually. So it's a mix of maybe 6 different citrullinated peptides that you test in a normal ELISA. And this is what is being used in clinics, but no one really knows the sequence of these, and it makes it difficult to kind of make further investigations in this. And here's just an overview of how the disease development looks in RA. So first you have a period where you might have some genetic risk factors, and the major one is the HLA, DR4. This is the major risk gene. And then you have some environmental risk factors, so this includes smoking, but also infections probably play a role. and it's believed that infections in mucosal tissues, so this is the mouth, the lungs, and the gut, that this might actually be the site of origin where a local inflammation leads to some kind of autoimmunity, where you have a formation of these ACPAs. But they can actually be present up to 15 years before disease onset. You can actually detect these ACPAs. So a lot of people are going around here with ACPAs, and they are completely healthy. But then something happens in a second phase, and this is when you actually get the symptoms. And what most likely happens is that you have antibodies that are specifically targeting the joints. And this is, for example, the anti-collagen antibodies, but there might also be these ACPAs that have started to target the joints, and this is when you get the symptoms. And then there's a third phase, and this is the phase when you actually have established RA and when you start the treatment. So the patient will often realize that something is wrong quite some time before they get diagnosed by the doctor. Thankfully, today we have some quite powerful drugs that can prevent this really chronic destruction, but still there is no cure for this disease. It's lifelong. So why do we need a new diagnostic test? I mean, we seem to have a good biomarker, we have really new powerful drugs, so why do we need a new diagnostic test? So there are some improvements that can be made. First of all, we would like to diagnose people as early as possible. And as I mentioned, it's not all of the patients that have these ACPAs. So around 30%, they lack a biomarker, or they lack a specific biomarker, which means that it will take longer time for them to get on an appropriate treatment. And during this time, you will have degradation of the cartilage. So one effort is to try to find new biomarkers to find the seronegative patients. Another thing is to try to predict the disease or predict the treatment outcome. So today, every single patient with RA that walks into the clinic will essentially leave with the same medicine. It's called methotrexate, and it's a very old drug, and it's quite efficient. And then they will test it for a few months, see does it work. Okay, if it doesn't work, then you get this anti-TNF inhibitor. And if that doesn't work, well, then you get another one. You might get an IL-6. If that doesn't work, we'll try CTLA-4 or anti-B-cell treatment. Essentially, but the problem is that we have no way of predicting who will respond to what drug. So we have lots of really powerful drugs that cost up to 15,000 euros per year in treatment cost, but we don't know when to use them. So this is a huge knowledge gap, so to speak. And another possibility would be to kind of try to predict RA in healthy individuals, because today, even though we can screen the entire population and say, hey, you there, you got ACPAs, maybe you will develop a disease in 10 to 15 years, but it's not ethical to say that unless we can tell them more definite when they will develop the disease. And so I'm not doing this alone. So this is the Joint ID Consortium. It was founded after we got a big grant from the Swedish Foundation of Strategic Research that had identified biomarkers in RA to be a very big knowledge gap that need to be filled. And so the group is led by Professor Rickard Holmdahl at the Karolinska, that is responsible for the basic research and finding the new biomarkers. We have Inge Gjertsson in Gothenburg, that is a clinician that is responsible for the contact with the patients and recruiting clinical cohorts. And then we have Jan Kihlberg in Uppsala, that is an organic chemist. He's the one that is synthesizing these triple helical peptides that are quite challenging to make. And then this is kind of like a big overview of the kind of test. So first, it's basically 3 components to it. First, we need the cohorts. So we need big cohorts of RA patients, pre-RA, osteoarthritis, psoriatic arthritis, lupus. And then we need peptides. So I already described this triple helical type 2 collagen, but then we also have some other novel peptides. And then technology-wise, in the original application, it said, we'll do everything with ELISA. And after the grant was approved, we kind of started calculating how many ELISAs we were going to run, and we saw, maybe there is a multiplexing solution that could help us. So we got in contact with the autoimmunity profiling facility at the Science for Life in Stockholm, and this is under the KTH University. So Peter Nilsson is the facility director, and he has a big, it's kind of like a core facility where they have set up a lot of different assays to do really broad autoantibody profiling. And in particular, they also have the Luminex. So they have 2 xMAP 3D machines and some other machines, and it's a really fantastic setup. So we got help there to set up our assay. And here are some different collaborators that have been giving us clinical samples. We have 5 different sites in Sweden, 2 in Germany, and also in China. And there are also some more coming in the future. And our current peptide library, it's a bit over 400 peptides. So it's, the core is kind of based on the triple helical type 2 collagen peptides. But then we've also identified 5 other joint proteins that we think are critical for the development of arthritis. And we're not disclosing them today, but this is based on a lot of functional animal research. That's to really get antibody or peptides that are relevant for the development of arthritis. And then we also have an important notice that all of them also have a biotin label. I will come back to that, why we need that. And then we have also some proteins, and of course we have a lot of the control peptides from the literature to see, can we beat what's the other ones on the market, kind of. So I will just quickly describe how we set up the assay. So first we have nutravidin coupled to the magplex beads, and this is the standard amine coupling reaction. And then we take our biotin-related peptides and add to the nutravidin beads. So then here we have a nutravidin coupled bead, and then we have biotin, and then we have a lysine knot that is connecting the 3 arms of the homotrimer peptides. And then we have used up to 360 different peptides in the same assay. And I was quite impressed that this actually worked, that we could put on 360 different peptides. And so I'm very happy that worked. And of course, before doing that, we also did some smaller experiments to optimize and also verify against ELISA, and it matched very nicely. And then for the data collection, so basically we have our serum, we mix it with the beads, we wash, and then we add a PE-labeled secondary antibody, and this can be exchanged, for example, for a anti-mouse or anti-rat for analyzing some animal samples. And then it's normal detection. And an important note here is that so we only require 1 microliter of cerat per assay. And most of our material, or essentially all of it, is coming from biobanks. And some of these biobanks have been collected for the past 40 years. And if we were to screen this on ELISA, we might need, if we want to repeat experiments, we might need half an ml of sera. And if you are contacting someone with 30-year-old samples and say, hi, could we get half a milliliter of all your patient samples? They would just laugh at you. But now we can say, maybe you can spare 20 microliters. That's all we need. We need no more for the entire project length. And so far, all that we have contacted have been really happy to supply this amount. And we know that people requesting larger amounts, they often get denied because they simply cannot spare the really precious samples. And here are the clinical samples we run so far. So it's 6 cohorts of RA, and then we have some other inflammatory joint diseases such as lupus, where some of the patients have joint symptoms. We've also tested a cardiovascular cohort because cardiovascular is the main comorbidity for RA patients. They actually they have a lower life expectancy, and this is due to cardiovascular disease. So we want to see if there might be a connection there. And then we have some pre-RA, juvenile idiopathic arthritis, osteoarthritis, relapsing polychondritis, and a few psoriatic arthritis. So now I will kind of try to give you some data on what we have done so far. So I'm dividing this into some different sections. Unfortunately, I don't really have time to go in describe these three, but I'll just touch upon the first 3 points. So the first is the
You have 10 more minutes.
So first is a diagnosis of established RA. So here is one of our new biomarkers, and I plotted it against 2 of the most well-characterized peptides in literature. One is called CSP1 from filaggrin, and one is CEP1 from alpha-amylase. And you see that there's a big overlap between the peptides, but we kind of uniquely can identify quite a few patients with this new peptide. Another peptide, also citrulinated, we kind of ran it head-to-head with this CSP2 test, which is the commercial ELISA that is today used in clinics, and we get almost the same sensitivity using one single peptide. And for the cutoff, we are using something called MAD, so it's a median absolute deviation, and we found that this is the absolutely best way of setting cutoffs. Standard deviation, don't use it. Search for median absolute deviation. And this is not MFI, this is actually, we're using a ratio, so for every sample we include kind of control peptides. So then we use a ratio for every patient between the negative control peptide and the peptide of interest. And that's what's being displayed here. And then, of course, that's what's only one peptide. What happens is we multiplex. So here we combined 18 different peptides, and we show that we could find almost 30% of the ones that are currently lacking a specific biomarker. We could actually find almost 30 of them. And then we miss out on a few of the ones that could be detected by the commercial kit. But overall, it's a major improvement to what's on the market today. And then something about disease severity. So here we looked at the disease severity at the time of diagnosis, and we correlated it to our peptide library. And here's one example or one biomarker that if we divide the patients into 3 groups, we have the ones in remission, low disease activity, and high disease activity. Here's an example of a peptide that among the high disease severity group, almost 10% of them are positive, whereas in the remission group, it's only 2%. So this is, and this has also been studied functionally in a mouse model to show that these antibodies are really pathogenic. And this is, of course, interesting to continue working with. And then something about treatment outcome. So here's another biomarker where we analyzed the patient sample at onset, and then we looked 6 months in the future and saw how have they responded to treatment. And here's an example of a peptide where in the non-response group, we have almost 10% of the patients, whereas in the ones that respond well, it's only around 3%. And of course, then the big question is, maybe these patients that are positive for these biomarkers shouldn't receive the standard treatment. Maybe there's another treatment that will work much better. And we don't know which treatment that would be, but definitely what's being given to them today is not working very well. And this is only one biomarker, and what we're struggling with now is trying to combine different biomarkers to make kind of model. And we also have biomarkers that are predicting for a good prognosis. And this is very challenging how to put together the data. And the kind of future plans right now is that we're going to do a technical validation of our top 50 peptides together with the Research Institutes of Sweden, a notified body. And so they will be done both in our lab and their lab to try to really make sure that we can technically validate them. And then we're also going to do a clinical validation of these 50 peptides in larger RA cohorts where we have a bit more statistical power. And then we're going to try to build multivariate models to try to make something that a clinician could read. Because if you tell a clinician, if you show them this slide, which I've done, they're like, what does this mean? So we need to give them a way to interpret the data. And one way to illustrate, because sometimes I get a question like, so couldn't you do this with ELISA? And one way to kind of illustrate this is that with ELISA you get 96 data points. So far in this project we've generated around 1.5 million data points. And this is the equivalent of over 15,000 ELISA plates. And if we put 15,000 ELISA plates on top of each other, it's 1,500, well, it's 1,600 meters almost, which would be the tallest man-made structure on Earth. And I'm also really happy not having to bring those to the recycling room. So this is kind of like a one slide just showing how much resources we save by doing multiplexing. And then, of course, here I'd just like to acknowledge some people that have been directly involved in providing the data for today's presentation. Thank you all.